Let me move this out of my way a little bit. Okay, for both the autonomic, both branches of the autonomic nervous system, first of all, the branches are parasympathetic. Well, this will not do. I want to use my stylus, and you are being a pain in the butt. Not parasympathetic, para. Hold on. I'm not going to use the whiteboard through Zoom. It's not liking my stylus. I'm going to go back to my PowerPoint and start a new PowerPoint. That communicates with my stylus a little bit better. Okay. That should be better. Okay. So let me make sure I have my pens up. Put your pens up. There we go. There are two branches of the autonomic nervous system. There is the parasymp. My computer just went, red alert, gonna shut down, give me a battery source. There we go. There is the parasympathetic division.
Yes. And the sympathetic. And
the sympathetic.
And that's all I know. No, I'm just kidding. I'm just kidding.
Well, I hope you have another way to describe the sympathetic.
Isn't that the fight or fright? That's the scary one.
Right. So normally students say fight or flight.
Yeah.
But it's actually 4 Fs. Fight.
Oh.
Flight, which means to run, to flee, right? You never know what kind of person you're going to be truly in a sympathetic moment. And most of you are going into the healthcare profession. And that means for most of you, you are going to have to train yourself to run towards danger, not away. And that means you're going to have to train yourself to go against the very core of your being, the visceral response of your being, to run towards danger. Most animals don't do that. They run from danger. They flight, right? They flee. So are you going to be a person that stands up and fights? Are you going to be a person that runs? You really don't know until you're facing that situation. Some people freeze. They're just paralyzed. They don't know what to do. And unfortunately, this even happens to nurses. There are some nurses during an emergency situation, they actually run away from the room where the patient is coding.
Wow.
And you can't find them. They're hiding somewhere. Some people, like I said, freeze, and they're just immovable. They're paralyzed for the most part. And then the 4th F is sex. So the 4 Fs. And by sex, I mean, of course, for an F word, fornication. Haha, you thought you were gonna get me. We say in anatomy, we say, forgive me, okay? But very difficult to draw this. Okay, point. This is supposed to be a sad penis. It's a sad penis. Point. P for point. So parasympathetic is in charge of erection and shoot. So point and shoot. So ejaculation is what I mean by the sex part.
Okay.
Yeah, the ejaculation part. So I'm going to erase my pornographic.
Drawings.
Yeah. Which, actually last Thursday, we got Zoom bombed last Thursday. I've never had that happen.
Oh, wow.
And it was during a meeting, and I don't know if they were actual Saddleback students, I don't know, but they jumped into the meeting and they took control of the meeting because the person who was host actually didn't have host rights. So she's frantically trying to kick them out of the meeting, and they start putting up horrible images of penises and vaginas, like hand drawing them like I just tried to. And theirs was they were a lot worse than mine, like anatomically incorrect. So I could not help myself. I kind of jumped in. I'm like, you should take my class. I can help you with that. I just couldn't help myself. Anyway, it was pretty awful, but we got through it. So that's what I mean by the 4 F's. The sex part is the ejaculation part.
Okay.
Parasympathetic, we say resting, digesting, peeing, and reading. All the things that you can do while you're quiet and at peace. I'm not saying that you should read and pee at the same time, but those are all things. If you're reading, you're clearly not running for your life at that time. If you're peeing, you're probably not running for your life at that time. There are some organs in your body, most of them, in fact, that are dually innervated by both divisions. Let's use your heart as an example. So the heart is innervated by sympathetic neurons, and it's also innervated by parasympathetic neurons. We would like our heart to know how to behave in each of these situations. Our sympathetic response is an alarm response. That's another way of describing it, alarm response. How does your heart feel when you're in this alarm response?
Fast.
Beating really fast.
Beating fast, and it's pounding in your chest.
Mhm.
You feel like it's coming out of your chest. Whenever your heart, the word for heart rate is chronotropy. And the word for the force of contraction of your heart is called inotropy. If your heart rate goes up, we say you have positive chronotropy. If the force of contraction of your heart goes up, we say you have positive inotropy. Clearly, during a sympathetic moment, you have positive chronotropic and inotropic effects in your heart. Does this make sense so far?
Yes.
Okay. In a parasympathetic situation, what do you think happens to heart rate?
Lowers.
Goes down. You're calm, you're resting, like when you're sleeping at night. So the parasympathetic division has a negative chronotropic effect. In both divisions, we have a 2-neuron chain. And the way I'm going to draw this 2-neuron chain, here's a circle. And that circle represents the soma, or cell body, the cell body of the neuron. The long line here represents the axon. And this split part represents the terminal boutons. A two-neuron chain, that means the upstream neuron is going to communicate to the downstream neuron. These are not dead stick figures I'm drawing. These are two neurons. There's an upstream and a downstream neuron. In anatomy, you learned about a better word to describe the upstream neuron. This functional contact between the upstream neuron and the downstream neuron is called a synapse. The upstream neuron in anatomy you learned to be the presynaptic. Guess what the second one was?
Post.
Good. Postsynaptic. Now, in anatomy, you also learned that if you have a collection of neuronal cell bodies inside the central nervous system, in the brain, it was called a nucleus. Not to be confused with the organelle. In the spinal cord, a collection of neuronal cell bodies was referred to as a horn. The dorsal gray horn, for example. Sometimes textbooks call it a column. My joke in anatomy was always, there are always two names for everything except for when there seems to be seven.
Okay.
So a dorsal gray horn, a dorsal gray column. When you have a collection of cell bodies like this neuron outside of the central nervous system, if it's in the peripheral nervous system, it was called a ganglion. So I'm going to start another page and redraw this fresh, and we are going to focus on the parasympathetic division. So again, we're focusing on the parasympathetic division tonight. Oops. And of course, it has a two-neuron chain. The first neuronal cell body is always found in the central nervous system, either in brain nuclei or in the gray horns. The dorsal gray horn, you've heard of that, the ventral gray horn. So it's either in nuclei or in these horns / columns. The second nucleus, the second cell body, soma, is always found in the peripheral nervous system, a collection of neuronal cell bodies outside of the central nervous system. Another way of saying that is a collection of neuronal cell bodies found in the peripheral nervous system is called a ganglion. So our goal tonight to review the anatomy is to identify exactly where are the cell bodies for the first neuron and where are the cell bodies for the second neuron. On the previous slide, we said the first neuron was presynaptic and the second neuron was postsynaptic. We can use better terms. We can use two other words that tell us we're dealing with the first neuron or the second neuron, and we're talking about the autonomic nervous system. The first neuron is called the preganglionic neuron. Why? It's before the ganglion, the collection of neuronal cell bodies in the peripheral nervous system. Care to guess what the second neuron is called?
Post.
Postganglionic.
Lucky guess.
You're on fire tonight.
The postganglionic neuron does synapse on something. It will communicate to something. The preganglionic neuron communicates to the post. And the postganglionic neuron is going to communicate to involuntary effectors. These involuntary effectors are heart, cardiocytes. You have no conscious control over your heart cells.
Thank you.
Smooth muscle. Smooth muscle is spindle-shaped and is mononucleated. Glands, and the way I draw glands is like this. Because most glands are exocrine, and the exocrine glands that we usually think of are coiled. So the parasympathetic division has this 2-neuron chain. Listen very carefully to my words. The preganglionic neuron is presynaptic to the postganglionic neuron. The postganglionic neuron, in turn, is presynaptic to the involuntary effectors. Did you listen how the words changed? Pre and postganglionic words merely referred to the 2-neuron chain. Preganglionic is the first neuron, postganglionic is the second. The word presynaptic in my sentences merely refer to which cell was upstream from another cell. The preganglionic neuron is presynaptic to the postganglionic neuron. The postganglionic neuron is presynaptic to the involuntary effectors. I'll say it again in layman's terms. The preganglionic neuron, the first neuron in our 2-neuron chain, communicates to the postganglionic neuron. The postganglionic neuron, the second neuron in our 2-neuron chain, communicates to the involuntary effectors. Now, how do neurons communicate? This is where, Marianne, I asked you all to watch the action potentials, membrane potentials, because in that video, you learn that neurons communicate through electro and chemical means. The electrical way is the action potential. It then is turned into a chemical signal, the neurotransmitter that is released. So now I want us to redraw this 2-neuron chain. And now let's draw what neurotransmitter is released from the first neuron. What chemical signal is released onto the second neuron for this parasympathetic system? And the answer is acetylcholine. Are there some exceptions? Of course, there's always an exception. But for the most part, we're talking about acetylcholine. Now, And that's what I want you to know for testing purposes. Acetylcholine, that's it. In the second neuron chain, or the second neuron of our 2-neuron chain, what is the neurotransmitter released onto the involuntary effector, say on the heart? It is again acetylcholine. Are there exceptions? Of course. Are there exceptions that I might talk about in my lecture? Yes. In fact, in a man's penis, the neurotransmitter that is released, which cannot be stored, it's made on demand, is nitric oxide. I feel like I'm writing no penis, but what I mean is nitric oxide is released onto the smooth muscle surrounding blood vessels that are found in a man's penis. When nitric oxide is released, it relaxes the smooth muscle around those blood vessels. And that is the reason why they become engorged with blood. They are less vasoconstricted, they are vasodilated, and the man's penis becomes erect. Okay, so you will actually hear about me talking about nitric oxide, and you will also hear me talk about a certain toxin from the Brazilian wandering spider. It's kind of an interesting, funny story. And you will also hear me talk about how some pharmaceutical companies are trying to combine the toxin from the Brazilian wandering spider to a current drug you might have heard of, Viagra. And they're trying to make a super duper erectile dysfunction drug. I have yet to hear if they've done this. It'll be interesting if they do, but you're going to hear me talk about nitric oxide and the Brazilian wandering spider. Okay, so how do I remember that for the most part, both the pre and the post-ganglionic neurons release acetylcholine? I remember the word parasympathetic, and the first two vowels are A's. And that helps me remember that the pre and post ganglionic neurons release acetylcholine. Again, is there an exception? Yes, the man's penis. That will be our notable exception for the parasympathetic division. Okay, the next part we need to piece together. The next part we need to piece together is what kind of receptor binds to this acetylcholine. So clearly, when the first neuron, the preganglionic neuron, releases acetylcholine, this diffuses across the neuronal synaptic cleft and will go to receptors found on the postganglionic cell body. There are different kinds of acetylcholine receptors. There are some called nicotinic acetylcholine receptors and muscarinic acetylcholine receptors. The way they behave, their physiology is very different from each other. That you are going to learn in your dendrites to synapse lecture. You are going to learn that the nicotinic acetylcholine is ionotropic and that the muscarinic acetylcholine receptor is metabotropic. Now is not the time that I'm going to go into those words and how those receptors work. This is merely a review of the anatomy, and we're trying to get an idea of how these neurons are communicating with each other. Okay. All right, so these receptors on the postganglionic neuron are nicotinic acetylcholine receptors. They are ionotropic. Ionotropic receptors are some of the most simple. Their physiology is easy to understand. Metabotropic receptors, second messengers from hell, baby. And it's a step-by-step process. It comes down to memorization, but it is a step-by-step memorization process. Now, what about the involuntary effectors? Clearly, they need an acetylcholine receptor. After all, the postganglionic neuron releases acetylcholine onto the involuntary effectors. What kind of receptor do they have? They have the muscarinic variety. They have the muscarinic variety, the more complicated receptor. When we get to the sympathetic division on another night, you will also realize that the preganglionic neuron for the sympathetic division also releases acetylcholine. You will also realize that the postganglionic neuron also has a nicotinic acetylcholine receptor. When we start to think that the heart is innervated by both branches, we should be getting a little nervous that both branches chemically start out the same. They both release acetylcholine from the first neuron onto the second. And the second neuron has the same kind of receptor for both divisions, nicotinic acetylcholine receptors. We should be nervous because if we're in a fight or flight response versus resting and digesting, we would like our heart to know how to behave. Heart rate and force of contraction should be elevated in one, and heart rate should be low in the other. And yet chemically, both divisions start the same. Not to fear, because when we get to the second neuron, you're going to realize that the two divisions use different neurotransmitters, and also the involuntary effectors use different receptors. And this is where the cells of the involuntary effector will be able to figure out, clear as day, which division is speaking to it, which division should be gaining more control. Are you in a fight or flight? Then the heart should behave like you're in a fight or flight response. Are you in a resting and digesting moment? Then the heart should behave in that way. No one would want to try falling asleep with a pounding heart in their chest. That would not be comfortable. We've had nights like that. They're sleepless nights, and we have a lot of anxiety, and our heart rate gets up, and we're just a nervous mess. It's not conducive for resting. It's awful. It's an awful feeling. All right, so now we are going to be in the central nervous system. This is where our first neuron is found. So I'm just going to draw a box to represent the central nervous system. Where are we? We're either in the brain, and forgive my sad drawing of the brain. There's the cerebellum. There's the midbrain, the pons, and the medulla, the brainstem. Okay, sad. It's a very sad brain. There are 4 places in the brainstem, 4 nuclei, where we find preganglionic centers for the parasympathetic division. Those four go to cranial nerve number 3, cranial nerve 7, cranial nerve 9, and cranial nerve 10. This is also found in the drawings that I did post under lecture unit 2. So I'm going to pick up the pace because we have about 15 minutes left. Cranial nerve 3 is called the oculomotor nerve. Cranial nerve 7 is the facial nerve. Cranial nerve 9 is the glossopharyngeal, and cranial nerve 10 is your vagus nerve. Cranial nerve 10 is the only cranial nerve out of the 12 that actually extends down into your thoracic and abdominal cavities. All 3 of those cranial nerves have preganglionic fibers in them. All of those nerves have preganglionic fibers. Cranial nerve 3 has preganglionic fibers. So does cranial nerve 7, 9, and 10. Be careful with these numbers. Cranial nerves 3, 7, 9, and 10 sounds dangerously close to other numbers you had to memorize for lecture unit 1.
Yeah.
Like clotting factors, 2, 7, 9, and 10. So cranial nerve number 3 is preganglionic, and it's going to communicate. I'm just going to draw it like this. It's going to communicate to a postganglionic neuron. And that postganglionic neuron is going to make things happen that you have no control over. Like it's going to go to the iris, the smooth muscle in your iris. And in your eye, so here's your pupil, and here's your iris. There are different fibers. There are different smooth muscle fibers. There are some that are circular, sphincter muscles, and then there are some that are radial, like spokes on a bike. So it turns out that cranial nerve 3, the preganglionic fiber, will communicate to a postganglionic neuron that communicates to the sphincter muscles. And when that happens, your pupillary diameter decreases. Cranial nerve number 3 is important when you're trying to do up-close reading. Of course, that's a resting activity. When you're calm, you can read. And when you're doing up-close reading, we say that you are experiencing accommodation, up-close reading. So not only does cranial nerve 3 and its postganglionic neuron communicate to the iris of your eye, it also communicates to the smooth muscle called the ciliary body that controls the shape of your lens. So not only do your pupils decrease in diameter, but your lens changes shape so you can do up-close reading. Cranial nerve 7 is also preganglionic to a postganglionic cell, and it's ultimately going to go to your sublingual and submandibular glands. So it's going to go to glands, and you produce more saliva. Cranial nerve 9 also synapses on a postganglionic neuron, and that's going to go to your parotid glands. Cranial nerve 10 is also preganglionic, and it will synapse on postganglionic neurons that go to your heart and your thoracic cage. Smooth muscle associated with your lungs for air passageways and regulating how dilated they are or constricted. It also goes to smooth muscle that is around your intestines. So if cranial nerve 10, your parasympathetic division, has more control of your gut, you have increased gut motility. You're going big potty. You're resting and digesting. Now, there is also not only these 4 brain nuclei, but also in the spinal cord, down in the sacral spinal cord, we have gray horns. Redraw that. No one should ever want me on their team for Pictionary, ever. So there's my central canal where cerebral spinal fluid is going. And here's a dorsal horn, and here's a ventral horn. And this is the middle, intermediate. And in the sacral spinal cord, this lateral horn in between the dorsal and the ventral has a very special name in the sacral spinal cord. It's called the sacral lateral gray column. Remember, sometimes horns are called columns. So the sacral lateral gray column is this middle horn. And again, the cell bodies that are found there are preganglionic. I'm drawing my preganglionic fibers in blue. They are going to terminate synapse on postganglionic cell bodies. And those postganglionic fibers will terminate on involuntary effectors. Not on the heart, because we're way down in the pelvic bowl, but they will synapse on smooth muscle. They will synapse on, say, involuntary effectors like, for example, the smooth muscle lining the corpora cavernosa, the erectile bodies in a man's penis that I referred to earlier. Oh, stop bothering me. So this is our review of the parasympathetic division. Now, in the pictures that I drew for you that are posted in Canvas, I want to explain you've got 4 pages. The first page is showing you the central nervous system portion of my picture I've drawn here. And I tell you that the parasympathetic division is called craniosacral in origin. Meaning, we've got preganglionic fibers that emerge from the brain, the cranium, and we have preganglionic fibers that emerge from the sacral spinal cord. That's the sacral part. Craniosacral in origin. And I draw these preganglionic fibers on page 1, and I tell you that they all release acetylcholine. Every single one. That's what you're going to see on page 1. On page 2, you are still seeing the parasympathetic division, but on page 2, I'm drawing the postganglionic fibers. And on page 2, you are seeing that I drew that the postganglionic neuronal cell bodies use nicotinic acetylcholine receptors. Also on page 2, I show that the postganglionic cells synapse terminate on the involuntary effectors. And I show you that the involuntary effectors use the muscarinic acetylcholine receptor. And I review that cranial nerve 3 with its postganglionic partner, remember it's a 2-neuron chain, will allow you to do up-close vision and decrease pupillary diameter. I also tell you cranial nerve 7 with its postganglionic partner will target your salivary glands, specifically submandibular and sublingual. Cranial nerve 9, the glossopharyngeal, and its postganglionic partner will target your parotid glands. You will have more saliva production. If you're resting and digesting and you smell a good meal, you're like Pavlov's dogs. You start salivating at the mouth. Cranial nerve 10, you'll see on page 2, innervates involuntary effectors in the thoracic and abdominal cavities. I tell you about how cranial nerve 10 and its postganglionic partner together will tell the heart rate to drop. It will tell the smooth muscle in the lung air passageways to contract. So you have bronchial constriction. When you're resting, you don't need as much airflow. And in the gut, cranial nerve 10 and its postganglionic partner will increase gut motility. You're going big potty. You're digesting. Things are moving and grooving in your gut. For the sacral lateral gray column, those preganglionic fibers and their postganglionic partners, they are going to target things, smooth muscles say, found in your bladder. And this is going to lead to micturition. When you're resting, that's when you can pee. Hey, have you ever tried to pee when someone's watching you? Do you get a little stage fright? It's a little difficult, right? To relax and allow your detrusor muscle to contract and allow the urine to be expelled from your body. And you're also going to see on the second page the erection story. And I have a little note saying, remember that's nitric oxide that is released from the postganglionic neurons. So down here, be ready to read acetylcholine is released, or in the case of the man's penis, nitric oxide. Again, I'm not saying no penis, I'm saying nitric oxide for the penis. Now, these postganglionic collections, these postganglionic neuronal cell bodies have a name. Like these 4 nuclei in the brainstem have names. They have names that you probably didn't have to learn in anatomy. Names like superior salvatory nucleus, inferior salvatory nucleus. They have names, these nuclei. In the sacral lateral gray column, that's the name of it, sacral lateral gray. There are, of course, anatomists like to give names to things. It takes too long for me to say, Sarah, the postganglionic neuronal cell body for the parasympathetic division. That took too long.
Mm-hmm.
So anatomists said, let's give these postganglionic neuronal cell bodies of the parasympathetic division a name. And they did. So all of these cell bodies that are postganglionic neuronal cell bodies for the parasympathetic division that I'm highlighting in blue, they called them terminal ganglia.
Well, that's not too bad.
Nope. But see, the parasympathetic division is easy. It's simple. This is not going to be the case for page 3 and 4 when we review the sympathetic division. It's a nightmare. It's much more complicated. The chemistry is complicated. There are different neurotransmitters released by the postganglionic neurons. It depends where you are. To make things even worse, the sympathetic division uses very different receptors on the involuntary effectors. They use adrenergic receptors. Adrenergic sounds like adrenaline. Adrenergic receptors would bind to adrenaline, also known as epinephrine. And there are different kinds of adrenergic receptors. There's alpha 1, there's alpha 2, there's alpha 3, there's beta 1, beta 2, beta 3. I mean, there's all sorts of receptors. Fortunately, I'm going to reduce the number of adrenergic receptors we ask you to know. Oh, and there's subcategories too. So we are going to ask you to know, we're going to ask you to know beta 1, for example. How many hearts do you have? Did you guess 1? Good. And what does your heart do for you? Does it beat? Yes, it does. So I think of beta 1 and my one heart that beats for me, B for beat and B for beta 1. That is the receptor we will find on the heart to increase heart rate and force of contraction. So, whew, thank goodness the heart, although it's dually innervated by both divisions, and both divisions will start out chemically looking the same, by the time we get to the postganglionic neuron, we're going to have different neurotransmitters released and different kinds of receptors on the involuntary receptors or involuntary effectors. And that means the lines of communication don't get crossed. The heart will know how to behave in a fight or flight response, and it will know how to behave in a rest and digest response. That is the end of our parasympathetic anatomy review. Okay. So, let me see your beautiful faces. Do you have any questions for me before I say good night and do one more professor-student conference for the night? Michaela, I don't know if you're still there, but I did reach out to you and I didn't hear back from you. So you and I are going to schedule for another time, if that's okay. I'll reach out to you again. Maybe we can do it in the next couple of days. Sarah, you and I are meeting tonight after class. Are you still okay with that? Where are you?
Yeah, I'm fine with that.
Okay, okay. So you and I are up next once I say goodnight to all of these wonderful people. Do you have any questions for me? And if you have, okay, and if you have reached out to me, trust me, I am not ignoring you. I am slowly picking through my list of students, trying to get to everyone. These conferences take about 30 to 40 minutes, depending on the student. And I'm trying to do about 5 to 6 students a day. So that's quite a bit of chunk of time.
Yeah.
In addition to all of the other professor type jobs that I have to do. Anyway, okay, if you don't have questions, then I'm going to say goodnight, and I will see you all Wednesday. We're talking about 2 physio-X's, so make sure you get those done if you can before we talk about it, even though they're not due until I believe I said Friday, right?
Friday.
Okay.
Bye. Thank you.
All right. Good
Bye,
night.
thank
everyone.
you. Good
Thank
night.
you.
Thank you.
Thank you. Oh, you're welcome. So Sarah, I'm going to put us in a private chat room so I can pull up your exam and go over your exam with you, okay? And then that way no one sees your business. So just give me a moment to pull that up and organize our little private chat room. There we are. Okay, dokie. Stop recording.